So I'll introduce now our first speaker today is Elizabeth Hanson. Elizabeth is a postdoctoral associate working in the lab of Benarenkiel at the Baylor College of Medicine. And she's new to the area of behavioral state regulation, and so she'd be really interested in hearing your feedback. Okay, go ahead.
Okay, I'll share my screen. Okay, is that good? Thumbs up from someone? Okay. So, hey, like Elaine just said, my name is Liz, and I'm going to tell you about work I've been doing as a postdoc in Benarenkiel's lab, Baylor College of Medicine. And in this project, I use a combination of fluorescent sensors, fiber photometry, and behavioral state manipulations to look at cholinergic signaling within the basal forebrain during olfactory-guided behavior. So I want to start by acknowledging the lab and especially my mentor, Benarenkiel. And I also want to highlight a super talented postbac, Katie Brandel-Encrup, who's helped a lot with this project. So I mentioned we're studying the basal forebrain, but that's actually a complex of nuclei, which each include a lot of cholinergic neurons, sending long-range projections to different brain regions. And these neurons mediate top-down control related to attention and arousal. They're important for cognition, learning, and memory. And their degeneration is associated with both cognitive and sensory deficits in neurodegenerative disease. So I'm going to focus on a region of the basal forebrain called the horizontal limb of the diagonal band of Broca, or HDB. And the HDB is especially interesting because it sends both cholinergic and non-cholinergic projections to the olfactory bulb and to multiple cortical regions. So it has a lot of potential to control both first order and higher order sensory processing. And by understanding this basal forebrain circuit and how it controls olfactory processing, we can begin to understand the role it plays in cognition and how its dysfunction contributes to cognitive decline. So we mostly tend to focus on cholinergic projections and their impacts on downstream brain regions, but there's also distinct roles for cholinergic signaling locally within the basal forebrain. So this is a diagram of some interactions between cell types in the basal forebrain. And I just want to focus on this interaction between the cholinergic and parvalbumin GABAergic neurons. So both of these populations in the HDB send long-range projections to the cortex and the olfactory bulb. But we don't really understand how the parallel projection streams work together to regulate downstream circuit function. We do have some specific clues about how this circuit might be working from another subregion of the basal forebrain, the medial septum. And the medial septum actually has a similar circuit motif of parallel cholinergic and GABAergic outputs, but targeting the hippocampus instead of the cortex and the olfactory bulb. And the upshot is that together, the cholinergic and GABAergic projections coordinate this oscillatory activity in the hippocampus. And at the same time, they set the excitability to a level where you get meaningful information nested within the oscillations. And so the key here is that cholinergic and GABAergic output is coordinated by this cholinergic signaling within the basal forebrain, which directly activates the GABAergic projection neurons. So from this, we hypothesized that GABAergic output to the cortex and to the olfactory bulb would be controlled by local cholinergic signaling within the HDB. And in this case, HDB cholinergic signaling would be a critical component of olfactory information processing, potentially even underlying coordinated sensory and cognitive deficits in disease. So this led us to ask the question, how does cholinergic signaling within the basal forebrain influence complex olfactory processing? And so we've approached this in two parts. First, by asking what actually is the pattern of cholinergic signaling in the HDB? And then what is the impact of HDB cholinergic signaling on GABAergic output? So to address this first question, historically, it's been pretty difficult to assay acetylcholine in vivo and in real time. So we took advantage of this new GPCR-based cholinergic sensor from Yulong Li's lab called GACH. And this allows us to directly measure changes in extracellular acetylcholine. So we injected a virus expressing GACH pan-neuronally into the HDB. And at the same time, we implanted a fiber optic over the HDB to measure cholinergic tone using a fiber photometry approach. And this allows us to directly assay patterns of cholinergic signaling in the basal forebrain in real time during freely moving behavior. So to get at the question of how these signals are tied to olfactory perception and decision-making, we imaged cholinergic signaling during the performance of an olfactory-based go-no-go discrimination task. And so the way this task works is that water-restricted mice are placed in a box with an odor delivery and a water reward port. And entry into both ports is monitored with an IR beam. And so mice are trained to self-initiate trials by poking their nose into the odor port, where they get puffed with one of two odors, S plus or S minus. And the S plus odor means that they can go to the reward port to get a water reward. The S minus means they need to reinitiate the trial. So once they're trained on the mechanics of the task, they can typically learn new odor reward associations within a single session of like 200 to 300 trials. So this flowchart just shows the possible trial progressions from odor presentation to the different potential outcomes. And as a mouse completes a trial, we can use the beam breaks at the odor and reward ports to determine when a mouse initiates a trial and when it seeks a reward. So using our knowledge of the trial outcomes and the timings of the port entries, we can infer the timing of certain behaviors and behavioral states within the task. So for example, trial initiation involves approach, active sensing, and odor perception. Then following odor presentation, there's a decision made and an action taken, either to seek a reward or reinitiate a trial. And each action comes with some expectation regarding the outcome. And finally, in at least reward-seeking trials, there's quantifiable feedback where they either get a water reward or they get a timeout punishment. So by combining this behavioral task with fiber photometry, we can look at patterns of cholinergic signaling in the HDB that correspond to these behaviors and behavioral state features within the task. So this is what the data looks like. These are individual trials from a single session aligned to the trial initiation times. And this is only showing the hit trials right now, but we can see a pretty clear pattern. So averaging across trials shows that as the mouse approaches the odor port to initiate a trial, there's a relatively flat baseline. But after the S plus odor presentation, the mouse is actively moving to the reward port. There's a sharp increase in HTB acetylcholine. Then once the reward is obtained, acetylcholine decreases again, and then actually falls below baseline some seconds later. And so this turns out to be a common pattern across animals and sessions in the HIT trials. And when you compare trial types, you see similar and different patterns emerge. So here, the trials are separated by outcome, notably in the false alarm trials where the mouse seeks a reward incorrectly. There's a similar increase in HDB acetylcholine between the odor presentation and the reward port entry. And so that's shown in these traces in the blue trace, that's the false alarms. So to quantify this across animals, I used the slope of the signal after the odor presentation. And the slopes of the hits in the false alarm trials are reliably higher than the correct rejects and misses, implying that this increase in cholinergic tone is related to the reward-seeking behavior. Another notable difference here between the trial types is this suppression, which is only present in the HIT trials. So to quantify this, I took the total area under the curve for the suppression below the baseline. And we found that the post-reward suppression is reliably larger in HIT trials, which indicates that the reward delivery is required for the suppression, not just reward seeking or reward port entry. So what I've shown so far with this data is that we can use fiber photometry and the GACH cholinergic sensor to directly record real-time changes in acetylcholine and the HDB during behavior. And doing this reveals that cholinergic signaling increases during reward seeking, which you can see in both the hits and the false alarms, and cholinergic tone is suppressed following reward delivery. And I'm just going to touch on the second part of this question briefly, which is about responses in the HDB GABAergic output neurons. So to begin to examine this, we used a similar fiber photometry approach, but instead of GACH, this time we're imaging GCAMP specifically in the HDB GABAergic neurons. And so in recording GCAMP fluorescence during the same go-no-go behavioral paradigm, we observed a similar bidirectional change in HDB GABAergic neuronal activity. However, this pattern is subtly different compared to the changes in acetylcholine. So for example, the increase in GABAergic activity after trial initiation is more tightly linked to the odor presentation itself. So in 3 of the 4 trial types, there's a relatively sharp response to the odor, and this is true for S+ and S- odors here. And where the response gets muddied is in the mist trials. So on average, the misses have much more variable and significantly longer times to peak. And so what we think this suggests is that in the mist trials, the odor wasn't appropriately detected or perceived. And this is directly in comparison to the false alarm type of errors, which require that an odor be perceived or detected but incorrectly identified. So these data imply that activation of HDB GABAergic neurons is tied to odor detection instead of reward seeking specifically. But then mirroring the change in cholinergic tone, we also find that GABAergic neuronal activity is suppressed following reward delivery in the HIT trials. So that you can see in the green trace here. And that's quantified here. So to end on, I wanted to come back to the main question and to this framework for understanding behaviors in the go, no go task. And I want to argue that by directly measuring HDB cholinergic signaling during behavior, we can begin to understand the role that it plays in olfaction and olfactory-guided decision-making. So far, we've shown that acetylcholine ramps up in the HDB between odor presentation and reward port entry. And because this happens in both HITs and false alarm trials, it indicates that cholinergic signaling follows reward-seeking behavior. And it may also reflect an internal state of expectation. So if this is the case, increased cholinergic signaling may play a role priming neurons in the HDB to respond to anticipated feedback signals, for example. And at the same time, we find that reward delivery transiently suppresses HDB cholinergic tone, suggesting that the signal is related to positive reinforcement. And intriguingly, the suppression below baseline also implies the presence of this baseline cholinergic tone, which can be bidirectionally modulated. And then when we look at GABAergic neuronal activity, on the other hand, we see a more general response to odors. And we think this is specific to active sensing because we don't see that kind of response with passive odor presentation. So the animal has to be like actively trying to sample the odor. But we also find that GABAergic neuronal activity transiently decreases after reward delivery, similar to what we found for HDB cholinergic tone. So we hypothesized at the outset that local cholinergic signaling controls HDB GABAergic projection neurons. And these data would suggest that cholinergic tone and GABAergic signaling are correlated, but only under specific circumstances during specific phases of the behavior. And the transient suppression of each signal seems to be involved in the positive reinforcement. And so I think this is surprising and pretty interesting because it implies some level of tonic activity in HDB GABAergic neurons, which is directly tied to changes in local cholinergic tone. And so we're working on teasing this apart now and looking at like GABAergic subtype specificity, projection specificity, that sort of thing. But that's where we're at now.
Excellent talk, Elizabeth. Thank you.
Thank you.
So we have a couple of questions. The first is from Christina. Which is the firing frequency range of cholinergic fibers? Are levels of cholinergic release correlated with increasing firing frequency of the fibers?
That's a really good question. And I think that depends on the target region you're looking at. So we don't actually know within the basal forebrain. Definitely that's true. If you look at downstream brain regions like the visual cortex, we know a lot more about cholinergic projections to the visual cortex. But I think that's a really interesting question to try and like dissect out the cholinergic release properties in the like distal brain regions versus the ones locally. So we assume that to some extent they're from the same neurons, but it doesn't necessarily mean that like a firing frequency at 10 hertz or whatever results in the same release properties in the visual cortex or in the olfactory bulb or within the HDB. So that's one of the reasons that we think the sensor work is so interesting because it lets us get at the actual like cholinergic release profile in different brain regions.
Great.
So yeah, the answer is we don't know.
So just one more question. It's from Alberto. He says, great talk. Is there an adaptation trial after trial in the timing or amplitude of the depression of ACH after the reward in HIT trials?
Yeah, so that is a really good question. And I didn't get to talk about any of the different sort of manipulations we did to the go, no go task. But the answer is, like, with just sort of baseline learning of an odor pair, no, there doesn't seem to be much of a change in the slope or the suppression, either the amplitude or the timing of the suppression. But when you start to get like weird changes in the amplitude or the timing of things, or when you do things like pseudo learning, which is where you basically do the same task, but the availability of a reward is not linked to the odor presentation. So you actually see like pretty dramatic modulation of the post-reward suppression in that case, which I think is super weird because they still get the reward, but they respond to it totally differently if there's not the odor reward association. And also like reversal learning, you see like pretty interesting changes in the temporal profile. But hopefully we'll publish this in like the next month or so, and that data will be in there.
Fantastic. Thank you, Elizabeth. So we'll thank Elizabeth once again, and thanks for the great questions.